Alright everyone, so I'm in Seattle and it's been fun. Hope you guys are having fun, not having class. Sorry about getting this out a little late. I am an idiot and forget that you guys are 3 hours ahead of me, so I clearly started recording this a little too late. But it's alright, you guys have the weekend to look over it too, so it shouldn't be too big of a deal. So today we're going to be talking about microbial genetics. Unfortunately, I don't have the ability to draw some pictures, so it might be a little bit difficult for some of the concepts, at least for me to explain them this way. However, I will include with this file a couple URL links to some YouTube videos that should help make some nice little pictures for you guys so it becomes a little more clear. Also, when I get back, we can do a quick little kind of synopsis of everything that I covered with this, especially some of the more, I don't know, conceptual stuff. So this is a somewhat long lecture. It's got about 80 slides. I'm going to skip over a couple bits and pieces here just so you guys aren't stuck listening to a movie that's an hour and a half long. Yeah, we'll see how this goes. Alright, so microbial genetics. So, okay. Can't do that, sorry. Alright, so a little bit about microbial genetics as a whole. So basically it's the understanding of DNA, and as we've learned thus far, DNA is basically the sequence for life. All the DNA within a cell is considered the genome, and little bits and pieces of that genome are considered genes. Genes code for any number of things, usually proteins, enzymes, all that good stuff. And the compilation of genes that a organism has is what dictates its life, basically. So it's good to think of DNA as kind of like a brain, but it doesn't have direct control over itself. The DNA itself is what dictates what happens within the cell, and it doesn't actually control itself other than that. Bacterial genomes are fairly small. They have anywhere from 0.4 to 9.4 million nucleotides. So a nucleotide is just an individual segment, or not segment, an individual molecule of DNA. Those are the A, T, C's, and G's that I'm sure most people have heard of before. And then you can see in the human genome, and most eukaryotes, it's much, much larger, about 4 billion nucleotides. And the majority of these are just junk. 90% of our genome is junk and it does nothing. We don't know what it is. It gets spliced out eventually. So it's usually or generally considered to be just carryover material from evolution. So you can see that our genome is about 1,000 times larger than E. coli, but it only has 6 times more genes, and that's because the bacterial genomes are basically just all genes, no junk. A little bit of junk, but not that much. And there's no splicing out, so it's just whatever's there gets read, gets transcribed, translated, all that good stuff. So you can see for E. coli, 85% of their genome code for protein, where in our genome we get about 2%. So it's definitely not a bad thing to have a super or small genome, especially if you're a prokaryote. And then we'll talk about the differences between genotypic and phenotypic. So genotypic is basically the sequence of an organism's genome. So that's the A, T, C, and G content. Where the phenotypic is what you can physically see, or what the organism is physically manifesting. So if you can sit there and sequence an organism and look at all the different genes, and you can say that because this gene has this sequence, this animal will have a phenotype of red hair or something like that. So phenotypic is much easier, obviously, to comprehend and to also get a hold of, or to analyze, because it's usually something you can physically see. But genotypic tends to tell you a lot more information, almost too much information. It's getting to the point now where we can genotype organisms, or genotype even ourselves or other humans, but now we have so much information, like the whole entire genome or genetic code, and we don't really know what to do with it. So technology really is kind of at the forefront of genotype research. So the induction of genetics and genes. So genetics is the study of the inheritance or hereditary of living things. So most organisms that have sex, like us, basically transmit our genes to our offspring, but at different rates. And for that reason, there is a natural variety between all of us. Things like bacteria or smaller prokaryotes we'll be focusing on mainly in this lecture go through binary fusion or binary fission, and they don't actually mix up their genomic material between a mate. Instead, they just make exact copies of it and transmit it to their daughter cell that they create. One way that they get variety, though, is by mutations. So bacteria are a lot more easier to mutate, where we have a lot more mechanisms that prevent mutations. So we have to rely on our ability to have sex to increase variability between our genome and our neighbor's genome. So it's basically the transmission of traits from parents to offspring, expression and variation of these traits. So that would be the phenotypic variety or the phenotypic expression of these traits. The structure and function of the genetic material and how this material changes. So it takes place in several levels, both the orgasmal, chromosomal, and molecular. So on the orgasmal level, you can see that's where the actual organism is composed of many cells, creating a tissue. At the cellular level, you're getting variation between individual cells based on their genetic material. On the chromosomal level, you're getting the variation between the different pieces of DNA. And then on the molecular level, stupid thing. Okay, on the molecular level, you're getting variation between the A, T, C's, and G content. So the nature of genetic material must be able to self-replicate. So if the genetic material can't actually replicate itself, the cell can't replicate. You need to be able to make a duplicate copy of your DNA so that when you create a new cell, it has the same genetic code as you did. And it must be accurately duplicated and separated from each daughter cell. However, things like prokaryotes and viruses tend to mutate much more readily than eukaryotes, especially viruses. Viruses are notorious for mutations, and that's one of their ways of evolving. That's how they tend to become super pathogens, such as HIV. So level and structure of the genome. So the genome is basically all the genetic material within a cell. Then you have bits and pieces of that genome where it's broken down into segments, and those are called chromosomes. And these compose of anywhere from thousands to hundreds of thousands of genes on each chromosome. And then to break it down a little further, you have genes, and genes usually code for either one or a handful of different proteins and enzymes. You can have things that code for structural things like actin, microtubules, all that good stuff. Genes for RNA machinery, so this is where it gets transcribed directly into things, or right into our RNA, which then can be made into ribosomes and such for making proteins. And then you also have regulatory genes, so you can have one gene that once it's turned on, it turns on a series of genes after it. And usually these genes are all part of a gene family, which code for something physiologically important for each of those genes, or they're all related somehow. So size and packaging of genome, so they vary greatly in size. Smallest viruses have about 4 to 5 genes. Again, you have to remember that viruses need to hijack a cellular's machinery, or the cell that it's infecting machinery. So it doesn't really need that many genes. It just needs enough genes to A, get it inside of the cell, and B, allow it to take over. So 4 or 5 are plenty. E. coli has about 4,288. That's because it basically lives a life where it doesn't require another organism. It doesn't need to take over cellular machinery, so it needs to have enough genes to code every protein that it's going to require to survive. And then human cells have about 20,000 to 25,000 genes, most of which don't really do anything, or we don't know what they do yet, but they are there. So the stretched out DNA can be 1,000 times more longer than the cell itself. So for that reason, we have to compact the DNA, and that's usually by wrapping them around histones. So in eukaryotic cells, going into chromosomes, so a chromosome is essentially condensed down DNA. So if you were to just let the DNA be loose and open, it would just take up the entire portion of the interior of the cell. So for that, we need to pack it down, and that's usually done by wrapping the DNA around histones in multiple different levels. So you have your helical DNA, which then gets wrapped around a histone 3 protein. Then those histone 3 proteins then wrap around another larger histone protein. And once that happens, you get this nice condensed down chromosome. Those you can physically see with your eyes under a light microscope, so they are fairly large. They're always located within the nucleus, and they vary in number from a few to hundreds. We have 46, but there are some yeasts out there that have, like, I think one species of yeast has like 2,000 chromosomes. So just because you have more chromosomes doesn't mean you're a higher order organism. It just means you've evolved and mutated slightly differently than another organism with smaller amount of chromosomes. They can occur in pairs like us, or haploid singles. So if it occurs in pairs, that means the organism reproduces via sex. So it has one set of chromosomes from mom, one set of chromosomes from dad. And each set of chromosomes has the same genes on there. The genes are just slightly different from mom and slightly different from dad, and whichever one of those genes gets turned on, usually by happenstance, that is what gene gets produced, and that's what it produces the person. And that's why people are unique. They're not the same as their parents, because it's a mixture of both genetic materials. Haploid organisms, on the other hand, are either gametes, or the actual reproductive cells of sexual organisms like ourselves, or they're asexual organisms like a lot of fungi and lower eukaryotes. And they appear to be linear, just broken up linear segments. Our chromosomes are in a nice little X shape held together by a centromere, but some higher order eukaryotes doesn't necessarily look like an X. Just depends on the organism. Bacterial chromosomes, on the other hand, they're condensed and secured by means of histone-like proteins, so they're not exactly histones, but they serve a similar function, so we give them the name histone-like. And it's a single circular chromosome, which is pretty easy to do if you have a small genome like a bacteria. So gene is a certain segment of DNA that contains the necessary code to make a protein or RNA molecule. So this is the bit that actually gets transcribed, translated into a protein. They're regulatory genes, so these control gene expression, and usually they're in front of a series of genes that they control. And sum of all types in an organism is called the genotype. So if you take all of the genes that are either turned on or turned off throughout a cell's life, that would compile the genotype. The expression of these genotypes create traits, and these are the phenotypes. These are the things that we can physically see, whether that is, someone's hair color, or going into a cell and looking at what proteins are created using molecular biological techniques. Both of them are considered phenotypes because you're looking at something after it has been transcribed and translated. And then all organisms contain more genes in their genotype than are manifested as a phenotype at any given time. And that's because our cells have the ability of saying, okay, we need these genes turned on at this point, but if conditions change or something drastically happens to that cell, then maybe another series of genes will be turned on. And this is all done by regulation, which we'll explain in a little bit. So the DNA code is just a simple yet profound message. So it was discovered in 1953 by Watson and Crick. It's basically made up of 4 nucleic acids and 2 strands combined into a double helical form. So it's this combination of all these, or these 4 different nucleic acids in different sequences, and this gives the wide variety of life we see today. So the general structure of DNA, the basic unit is called a nucleotide. So this is the A, T, C, and G by themselves. The basic backbone of a nucleotide is the phosphate, followed by deoxyribose sugar, and then the nitrogenous base. The nitrogenous base is what varies. That's what makes your A, T, C, and G. These are covalently bonded together to form a sugar-phosphate linkage, the backbone of each strand. One bond is to the 5' carbon on the deoxyribose, the other is to the 3' carbon. So this allows you to create a single or a strand of nucleotides. And these strands of nucleotides are what compose of genes. And because they have this nice pi covalent bond between them, it becomes very hard to pull apart. And this is what prevents mutations from occurring. Because if you have a nice sequence, and the sequence codes for a gene that you really, really need, if that sequence can be broken up by these bonds breaking, you then would have some sort of mutation, because you wouldn't get that gene being produced the way that it should be. That mutation would obviously be bad, because that gene is required for life. So it's important to keep these nice covalent bonds between these nucleotides so that the sequence doesn't get mushed up or switched. But it does happen, and it's usually because of forces that are strong enough to break these covalent bonds, such as UV radiation or heat, or some elements like oxygen or oxidizers. So here's a kind of a molecular picture of DNA. You can see the A, C's, G's, and T's. This is what composes the nitrogenous bases, followed by the deoxyribose, and then the phosphate bond. Here you can see these nice covalent bonds that bind the nucleotides together into a sequence, and then you have one strand here, and then you have another strand of the DNA here, and they're held together by hydrogen bonds. Now, hydrogen bonds are weak. They're the weakest bond around, basically, and for that reason they're pretty easy to pull apart. If you pull these hydrogen bonds apart, you can then make a copy of either strand of DNA, and the cell utilizes this to create identical copies of its genome when it's duplicating. You can see here between G's and C's you have 3 hydrogen bonds, and between T's and A's you have 2 hydrogen bonds. So it's weaker at the T and A bond site. So whenever you want to start duplicating your DNA and you need to kind of separate these 2 strands, it tends to happen at a TA rich site. And that's because these are weaker, and it's easier for helicase to get in and break them apart. So nitrogenous bases, you have your purines and your pyrimidines. They span the center of the molecule and pair with complementary bases. These pairs are called base pairs. Paired bases are joined by hydrogen bonds, which are easily broken and allow the molecule to be somewhat unzipped, allowing for DNA replication or transcription. Adenine and guanine are your purines. Thymidine and cytosine are your pyrimidines. And adenine is always paired with thymidine, and guanine is always paired with cytosine. So you have A to T and G to C, always. Oops. Antiparallel arrangement. So you basically have 2 strands of DNA, one running in the 5' to 3' direction, which is basically like reading from left to right, like we do now. And then you have another strand that's complementary to that, running in the opposite direction. So it's running from 3' to 5'. So it's running in a backwards direction. This just allows for a more stable